Hello and welcome back to Science of Medicine. Today I want to talk about fluidic compartments, in particular the aqueous compartments within your body, and how your body accommodates the need for water moving from one location to another. This is very important clinically because many times we will give fluids intravenously to address certain problems. Now, as always, when I discuss this, I will put a series of videos dealing with physiology up. And if you like this, please make sure you like and also subscribe to this channel. So in human physiology, I'm trying to understand and model mechanisms by which the human body is working. My overall goal in any kind of human life is to live and reproduce. But in order to do this, I have to have relatively constant internal conditions. I have to control for temperature so that enzymatic reactions can occur at a certain rate. I have to have certain ions and energy. I have to have a certain milieu in which the life of cells can occur and those cells can then reproduce. That milieu of these homeostatic conditions is essential. And since most of my chemistry is occurring within an aqueous compartment, this is very important to understand in understanding physiology. I also find that students have a difficult time understanding this, which is why I'm talking about it separately. Now, depending upon what book you read, we will look at total body water as being about 60% of your weight. That is, if you were to take all the water out of me, about 60% of my weight would go away. And this does vary depending upon the person. Some people will have more fat in their bodies than others, and so that will not be an aqueous component. Women in general will have a higher body fat content than men because they have to, if they're in reproductive age, accommodate the chance of having a child and another body inside them. And so they tend to have higher fat content because of that. And then, of course, people will be of different sizes, and so there'll be different body compositions. But typically around 60% of your body, if you are a male, is water. About 2/3 of this will be inside the cells at any one time. That is an intracellular component. And about 1/3 will lie outside the cells. Now, outside the cells is comprised of two different areas. The interstitium between the blood vessels and the cells is comprising about 25% of your total body water. Depending upon which physiology text you read, this may be a little bit different. And the plasma within your bloodstream is going to comprise about 8%. So 8 and 25 is your other 1/3 of your body water. Now, plasma also contains things like phospholipids and cholesterols and some fat. But in order to maintain this homeostatic environment, I have to be able to move water across this. That is, I have to be able to move water from the plasma into the interstitium and then into the cells, because I have various inputs into the plasma. Sure, I could have an intravenous way of actually putting that in, but most of us will input fluids in through the GI system. If I'm thirsty, I'll drink some water. And then I'll actually have fluids that will go outside, water which will go outside predominantly through my urine to be able to maintain that balance. In order to go from my bloodstream into the interstitium, I have to pass a membrane. That membrane is the capillary membrane of my blood vessel. And then that interstitial fluid is bathing my cells that are performing whatever function they're performing. If they're hepatocytes, then they're liver cells, they're performing their liver cell functions. And they have cell membranes, and I have to move that water across it. Now, fortunately, I have pores that allow for this very rapid movement of water, and those aquaporin channels will allow for this. But there has to be a driving force, and that driving force is osmotic pressure. So I move water inside my body across these different domains based upon osmosis. And that means that I have to have an osmolar gradient to move water. Now, if I don't have an osmolar gradient, then there'll be no net flow of water from one compartment to another. But if I do have a different osmolarity in the interstitium versus the plasma, then I can have a net movement of water. Now, it does take time for water to go from the input, my GI system, into my blood, and then from the blood into the interstitium and into the cells. And this might take several hours, for example. So let's talk more about this movement. In order for this movement to be spontaneous, as I mentioned, you have to have an osmolar gradient. Now, I'll talk a little bit about osmolarity and osmolality. I hope that you remember that osmolality is a measured quantity where you're measuring the amount of osmoles, or these particles that are dissolved in water that will hold on to water, relative to the kilogram of solvent, whereas osmolarity are these osmolar particles expressed per liters of solution. For our purposes, even though we measure plasma osmolality, because I use a freezing point depression method for doing this, and that measures an osmolality, I'll use the term osmolarity and osmolality synonymously. So osmosis is a net movement of water across this semi-permeable membrane, and it's spontaneous from a region of low osmolarity. That means that I have more water, to a region of high osmolarity, which is a low water concentration. This is always a difficult concept for people, because you go, how can you move from regions of low to regions of high? Isn't that opposite of what you would expect? It depends upon your perspective here. In this case, osmolarity is a measure of the osmolytes per volume of water. If I have a low osmolarity, it means I have a high volume of that water, which means I have a high water concentration, if you were to think about it that way. And likewise, for a high osmolarity, I have many more particles per volume than a low osmolarity. And so it's got a low water concentration. I wish, actually, that we would express this in terms of water activity or water concentration, but we don't. We use osmolarity, and that's sort of an opposite effect if you were thinking about it. Although clinicians don't generally calculate osmolarity, I will actually point out to you that to get this osmolarity, I'm going to sum up all of the particles that are dissolved. And I can actually use a formula for that, but I wanted to show you that the osmolarity is proportional to the concentration of those species. So if I have sodium chloride dissolved into sodium ions and chloride ions, then the concentration of sodium, concentration of chloride, each contribute to the osmolarity. In fact, this Ni is the number of particles that, or osmolytes, that a species will dissolve. So for example, sodium chloride in the body can be present as sodium ions, chloride ions, or sodium chloride that is aqueous, that is not actually dissolved. But sodium chloride will only have this Ni of 1 because it doesn't dissolve. Whereas if it dissociates, then the sodium chloride concentration will yield 1 mole of sodium, 1 mole of chloride for every 1 mole of sodium chloride. There is an ideality or non-ideality constant here, this form, which we won't concern ourselves with at this point, but there are drastic deviations from ideality. At this point, I'm not going to discuss that because that's getting into things that are much more advanced. So osmolarity is something that is dependent upon the concentration of dissolved particles in a certain volume of solution. It takes time to do this, and as I mentioned, if I take a bit of blood, I can measure the plasma osmolality we'll consider that to be synonymous with osmolarity. So what kinds of particles am I talking about? Well, it depends upon what fluid compartment you're talking about. So if you're talking about things such as the cells, the predominant osmolar particles are potassium, phosphate, and proteins. It doesn't mean that I don't have chloride or bicarbonate. I do. But the predominant concentrations, the ones that are the highest concentration inside the cells that are holding on to the water, inside the cells, potassium, phosphates, and proteins. Inside the plasma, it's sodium, chloride, bicarbonate, and albumin, albumin being the protein that's prevalent in the blood. And in the interstitium, it's sodium, chloride, and bicarbonate. So it's important that you keep each of these osmolytes in mind in the different compartments. Now, some osmolytes can move between compartments relatively spontaneously, and others have to be actively transported. Active transport meaning that I have to actually use energy to move from one compartment to another. And most of the time that's because I'm going against a concentration gradient. So these osmolytes can be small molecules, glucose, urea. They can be ions, as I've mentioned, sodium phosphate, potassium phosphate, chloride, bicarbonate, as well as proteins. The one that I mentioned in particular was albumin. Now this plasma osmolarity is very tightly regulated in the body. We have a set point in our brain of about 290 milliosms per liter. And I adjust this under the action of antidiuretic hormone, which is released from my pituitary gland. And when I release this antidiuretic hormone will act on the reabsorption of water in my kidneys, which we talk about in renal physiology through the aquaporin 2 channels in the cortical collecting duct, as well as stimulating a desire for fluid intake. So if the set point is 290 milliosms per liter, if my osmolarity in my plasma goes to 300 milliosms per liter, then I'm going to release ADH, and when that happens, I'm going to retain more water, hold on to that water, and stimulate a thirst response so that I go and drink some water to try to lower that towards 290 milliosms. If it's lower, if it goes down to 280, then I'm going to be able to excrete additional water and I won't be thirsty. Now, many times physicians will use fluids that we will infuse intravenously or into the plasma, into the blood vessels. And sometimes I characterize those fluids that are infused by the terms isotonic, hypertonic, and hypotonic. An isotonic fluid is one that has the same osmolarity as plasma. A hypertonic fluid has a greater osmolarity, and a hypotonic fluid has a lower osmolarity. Now, I will tell you that sometimes physicians don't speak precisely about this. So, for example, when I say an isotonic fluid, it may not have an osmolarity of 290 milliosms per liter, or maybe the person has an osmolarity of 300 milliosms per liter, and therefore the isotonic fluid would typically have a osmolarity of 300 milliosms per liter. What physicians generally refer to as isotonic is something that is somewhere close to 290 milliosms per liter. And that's the way that I'll actually use it in these slides. So isotonic, hypertonic, and hypotonic are fluids that I can give intravenously, and those have important implications. I mentioned that I can also transport things other than water, but these can be small molecules or they can be ions. I can have ion channels, for example, that transport ions spontaneously down a concentration gradient. I can have active transport via a Symport. A Symport is basically a transporter that transports multiple substances in the same direction. For example, in your gastrointestinal tract, there's a sodium glucose transporter, so that if I'm really dehydrated, I can use an oral rehydration solution that will allow sodium, glucose, and water to be transported in across my gastrointestinal tract and allows for quicker rehydration. Antiports also provide for transport of multiple substances, but in this case, they move in opposite directions. For example, there is a transporter in the loop of Henle, which transports a sodium, a potassium, and 2 chlorides that actually work as an antiport and move those ions in opposite directions. So let's talk about some applications because it's easier to understand this by working problems. So let's say you were asked to see an 88-year-old female. She comes from a nursing home and she's severely dehydrated. She's not been drinking or eating much for the past 2 days. Maybe she has an infection. Maybe she has not had much intake because she's demented and didn't realize she wasn't eating. When you examine her, her plasma osmolality is elevated at 300 milliosms per kilogram. So we'll consider that to be 300 milliosms per liter. And she's producing very little urine as appropriate because you would expect that antidiuretic hormone would be holding on to as much water as possible because she's dehydrated. So which of the following intravenous solutions would be best for rehydrating her? I could give her a solution of normal saline intravenously. Now, normal saline is 0.9 grams of sodium chloride per 100 milliliters of solution. We're going to consider this to be isotonic with plasma. Technically, it's not, but it's close. And in her case, it's actually not going to be because she's more concentrated. Sodium chloride will remain in the extracellular compartments, that is, in the interstitium in the plasma. I could give her D5W, which is dextrose, 5% in water. Dextrose is just glucose. And glucose will distribute evenly throughout all the fluid compartments. I could give her hypotonic half normal saline, 0.45 grams of sodium chloride per 100 milliliters of solution. And the sodium chloride also remains in those extracellular compartments. Or I could give her a hypertonic solution of 3% normal saline, which is 3 grams of sodium chloride per 100 milliliters solution, and the sodium chloride again remaining in the extracellular compartments. Now, I don't give her deionized water intravenously, and the reason for that is if I gave her that, then all the cells would swell up and they would lyse. So we don't give water directly intravenously. I can drink water, but I can't give that intravenously. So for this little old lady who's dehydrated, which of these solutions do you think is going to be the best to rehydrate her, to get water to move into all 3 compartments? So think about that for a minute. I hope you recognize that the D5W is going to be the optimal thing to give in this case. So dextrose or glucose is going to distribute between the intracellular, the interstitium, as well as the plasma. Because of that, the water is going to follow that in equal amounts, in equivalent amounts. So I'm going to be able to rehydrate her. On the other hand, the normal saline, which is isotonic, the sodium chloride is going to remain in the extracellular compartments only. That is, it's not going to get inside the cells, and so water isn't going to go into the cells as much because it's going to have that sodium chloride that's going to try to hold on to it in the interstitium and in the plasma. In a different way, hypertonic solution, 3% normal saline, is going to tend to draw water towards itself. It's not going to help with rehydration. Now, the hypotonic solution, half normal saline, will allow for water to move into the intracellular compartment. But because the sodium chloride is going to be holding on to that water, you could think about it as kind of being a normal saline solution with a solution of water or deionized water. So in this case, I could convince myself that half normal saline could be equivalent of normal saline, which is going to stay in the extracellular compartment, and deionized water, which will distribute between all of the compartments. So if I look at that, I'd have 500 milliliters of normal saline plus 500 milliliters of water. But in the case of the dextrose, I'm going to get whatever I put in there. So if I put a liter in, I'm going to get a full liter. So let's talk a little bit more about that. If I infuse 2 liters of D5W, how much water is going to remain in the plasma after that time? So remember that the plasma is going to comprise about 8% of my total body water. So if I have 8% of 1 liter, that's going to be 80 milliliters times 2, that's going to be 160 milliliters. Let me show this a little differently. If I look at after equilibration of the dextrose, I'm going to get 67%, or 670 milliliters of the 1 liter is going to go inside the cells. I'm going to have 25%, or 250 milliliters for every liter going in the interstitium, and I'm going to have about 80 milliliters staying in the plasma. So 8% of 2 liters is 160 milliliters. All right, let me do another one. Suppose I have a 45-year-old person who has a closed head injury that leads to the brain swelling. It's called cerebral edema. As a temporary measure, her neurosurgeon is going to order infusion of a solution that will draw water out of the intracellular space, in particular in the closed head area in the brain. That will help it so that as it swells, it maybe won't swell so much that it'll swell down the spinal cord and cause death. So which of the following intravenous solutions would be best to draw fluid out of the cells? Normal saline, D5W, the hypotonic half normal saline solution, a 3% normal saline solution, which is hypertonic, or albumin, which is isotonic with the plasma, but the albumin remains in the plasma. So hopefully you recognize that if you want to dehydrate the cells, you want a hypertonic solution, one that's going to draw fluid out of those cells, and so you would use the 3% normal saline. This is an important concept, so I hope that this helps answer some of your questions which relate to fluids within the body. I'll be back and talk to you more about science of medicine and medical physiology in later videos.